Another group of factors that affect enzymatic rate are molecules that bind to the enzyme and alter its structure and therefore its function. We call these activators and inhibitors. Here I'm going to focus on inhibitors, but activators function in the same manner, just kind of in reverse. Inhibitors fall into two major groups. One is a competitive inhibitor. A competitive inhibitor binds to the enzyme in the same area as the substrate. It binds somewhere within the substrate binding active site. Here we see our substrate binding site, and then the yellow is the competitive inhibitor. This molecule is able to bind to that active site, and when it's bound, it changes the shape of the enzyme so the substrate no longer fits in the substrate binding site. This is possible because of the structure of the competitor and the enzyme. The chemical structure is such that the competitor can bind. Sometimes this is a competitor that our body produces naturally. It's producing it to control the enzyme, to kind of keep it from going crazy. And we'll come back to this idea. Sometimes the competitor is a toxin and it's secreted by another organism. And so our body isn't trying to turn the enzyme off. It's maybe a microbe that is creating this toxin. Sometimes this inhibitor can be a medication. An example is the antiviral Tamiflu, which is a competitive inhibitor for the enzyme neuraminidase, which is a necessary enzyme for a virus to release from the host cell. So these could be a good thing. Non-competitive inhibitors bind to the enzyme, but in a location separate from the substrate binding site. The site where the non-competitive inhibitor binds, we call the allosteric binding site. So it's not uncommon to hear non-competitive inhibitors called allosteric inhibitors or allosteric molecules. When we compare the inhibition rate of these two classes of inhibitors, in general, our competitive inhibitor is going to take a little bit longer to inhibit your enzyme. What's happening is you're reaching saturation quicker, but you still have to reach saturation in order for it to inhibit the enzyme effectively, versus non-competitive inhibitors where you're pretty much turning that enzyme off. For both of these inhibitor types, depending on the specific inhibitor we're talking about, this binding can be reversible or it could be permanent. For regulation in a system like the human body or in a bacterial cell, within a system like this, you want your inhibitors to be non-permanent. You want them to be reversible. It would be beneficial for the system, you would like it, if this inhibitor was able to come off the enzyme and then the enzyme can work again. Having to create all new enzymes because the inhibitor is bound permanently takes time and energy and amino acids. It takes materials to do. So you would like to be able to use these enzymes again. So in body systems, we usually see the inhibitors are reversible. But in some examples of toxins, you see irreversible binding to the enzyme. For example, there's a chemical in nerve gas that is an irreversible competitive inhibitor to cholinesterases, an enzyme necessary in the synaptic cleft to clear array neurotransmitters. For a specific micro example, we have the antibiotic penicillin, which binds irreversibly to the enzyme Ch5pt3 found in the microorganism, and transpeptidase is required for cross-linking within peptidoglycan. So penicillin binds irreversibly to the enzyme necessary to cross-link peptidoglycan, meaning that peptidoglycan does not get cross-linked and you can't build a strong peptidoglycan cell wall. This is a great place to introduce feedback inhibition. I've kind of been talking about this as we went the whole time, but we're going to kind of consolidate this idea. Let's first walk through our system together. Here we have an enzymatic pathway. A pathway is a string of reactions that are tied together by different enzymes, and the pathways have this kind of passing of substrate to product, and then that product becomes the substrate for the next enzyme in the pathway. So let's walk through our pathway. We have substrate red that will bind to enzyme 1. Enzyme 1 has an allosteric binding site that we will come back to later. Enzyme 1 will act upon its substrate red, and create a product. Because this product acts as a substrate for the next enzyme in the pathway, we call it an intermediate. Intermediate is a word you hear a lot when we're talking about metabolism, because this is a very common situation we see in metabolism when we're talking about all of the biochemical reactions occurring in an organism. Intermediate A then becomes the substrate for enzyme 2, which acts upon intermediate A and converts it into intermediate B. Intermediate B becomes the substrate for enzyme 3, which then acts upon intermediate B to create our end product. This is the final metabolite from this pathway. This pathway is present to create this end product. Let's say that this end product is an amino acid. Amino acids can be used for a number of things in the cell. But you only need so many amino acids at any given time. You don't need 10 pounds of tryptophan. That's just crazy. So what feedback inhibition does is it allows this enzymatic pathway to function when the end product is required, when you need more of this amino acid, but it will shut down when you have enough or too much of the end product / amino acid. So let's see. We have enough of this amino acid. That's what we're saying here. We've gone through the pathway to create enough amino acid. What some of those amino acids will then do, what some of these end products will do, is they will bind to the allosteric binding site on enzyme 1. They're acting as a non-competitive inhibitor. They're acting as an allosteric inhibitor. When the end product binds to enzyme 1, acting as a non-competitive inhibitor, it changes the shape of the substrate binding site, and therefore this first substrate red, so it can't bind to enzyme 1, and it can't make intermediate A or B or end product. It shuts the whole pathway down. So that's how you prevent a pathway from creating too much of a product, of an end product. But what if you need to start making this amino acid again? What if you need this end product again? Well, what will happen is the concentration of that end product in the solution will drop as it's being used by the organism. When that concentration is low enough, the end product bound to the allosteric binding site will unbind, and it will allow this enzyme 1 to go back to its original shape and the pathway will turn on again. So this is an example of a reversible non-competitive inhibitor. I want you to think about how beautiful of a setup this is. This allows a cell to control how much of a product it has. So when we're talking about cells wanting this much, or it makes this much because it only needs this much, it doesn't require a brain to know when to stop. It's all based on physics and chemistry. It won't make too much because it'll shut the pathway down using feedback inhibition. It won't shut the whole thing down permanently so that you drop too low, because this is a reversible binding to the allosteric binding site. This is beautiful. And cells in general, all cells do this. And this is how the cell can control its metabolic pathways, or at least one of the ways it can control the metabolic pathways.